So I am delighted to introduce our speaker this evening. James Heal is the chair of the Wren Wildlife and Conservation Group, which is based around the Wanstead area in East London. He is a birder, amateur ceciologist, and a naturalist with an interest across taxa. James is a member of the British Plant Gall Society, and he's written several papers on galls, which have been published in their periodical. I'm going to stop sharing my screen now, and I'm going to hand you over to James.
Thank you very much indeed. Hopefully you can all hear me. I had a slightly scary moment there where I was kicked off for a few moments, so I missed some of your introduction, but hopefully I'm back on now and everyone can see and hear me. So let me share my screen, get that up straight away. And
yeah, just to let you know, that's fine, James.
Great, thank you, Maria, that's much appreciated. Okay, super, so thank you very much for that introduction, and incredible and a little bit intimidating to see so many people here, but really, really wonderful that there are so many people and such sort of a level of international interest in plant galls, particularly because I have to say my presentation focuses on plant galls in the local area to me, but I will, hopefully it is of relatively broad interest. So in the intro that Maria gave about me, you said that I was an amateur ceciologist. Yes, emphasis on the amateur, I suppose, in a sense. I would perhaps prefer to almost refer to myself as a galler, really, rather than a ceciologist, just as I generally prefer to refer to myself as a birder rather than an ornithologist. But, you know, I go out, I look for galls, I read about them, I do study them, so I kind of leave it up to those in the audience as to whether you consider me to be a galler or a ceciologist. So this presentation is going to be primarily aimed at, and so that was a helpful poll just now, it is primarily going to be aimed at people with relatively low, little, or even no knowledge of galls. So I noticed that there were possibly 3 people who consider themselves to be experts. Somebody has said they could write a book about it, and I imagine perhaps they may even have done that. And you will almost certainly, if you are anywhere close to that bracket, have more knowledge of plant galls than I do. So perhaps there are others who can join in the discussion when we have time for questions and discussion at the end. So that's just one thing in terms of sort of the I suppose the nature of this presentation. And the other thing I wanted to say is that this presentation is primarily organized around gall causes. There are a number of different ways that you can organize, I suppose, a presentation on plant galls, and I will come back to that a bit later. But this is primarily focused on galls categorized by gall causa, and that the main reason behind that is because from a wildlife recording perspective, and I'm sure there are many keen wildlife recorders on here, that is roughly how we focus on the sort of I suppose the definition of plant galls. So the only other thing I wanted to say right at the beginning is if you are relatively new to plant galls, you've obviously shown enough interest to join this talk, and I really hope my primary objective is to leave you with perhaps a thirst and an interest to find out even more about plant galls. I find them fascinating, very obviously, and partly because it allows you to study right across taxa. So, you know, it's an opportunity to get closer to botany, it's an opportunity to get closer to entomology, to mycology, to, you know, a number of other areas of interest. So let's jump in straight away with what is a gall, and I suppose what is not. So, and I should also say at this point that all the photos in this presentation are taken by me. So if there's sort of a relatively poor quality, that's that's because I've taken most of these with my camera phone and often with a small lens device attached to it, just to sort of get a bit closer to the subject. So these three images here kind of give you a sense of some of the things that you might find when you're walking around in the UK, I'm afraid, and although you will find some of these elsewhere. And not all of them are galls. 2 out of these 3 images are galls. So really, I just sort of want to give a sense of kind of what we're talking about in terms of what is a gall and what is not, and then we'll get a little bit deeper into the definition. So the first image on the left, you're effectively looking at a large growth on an acorn, on oak. In the second image, you're looking at effectively raised, enlarged, wavy veins on the leaf of hornbeam. And in the third image, you're looking at a discolored patch and sort of a bumpy patch on hollyleaf that I imagine is familiar to many people. And the first 2 images are plant galls, and the third image is not a plant gall. It's a leaf mine. But I just want to sort of touch a bit more on what is a gall. So we've got a nopper gall on the left, on the 2 nopper galls actually on acorns there, caused by a wasp. In the middle image, those raised sections contain an area where there are reasonably large numbers of mites, Aculops matrotrichus, inside the hornbeam leaf. And in the third image, these are leaf mines caused by the aggregate group of Phytomyza illicis. So let's just dig a little bit deeper into the definition of what a gall is. And there may occasionally be a very slight delay before I'm able to flick across to slides, but here we go. So what is a gall? This is the only page really where I've got some text on. And this is taken from UK purposes from perhaps the most important book that we have on plant galls. There's sort of a really useful guide on plant galls by Margaret Redfern and Peter Shirley. So a gall is an abnormal growth produced by a plant under the influence of another organism. It involves enlargement and/or proliferation of the host cells or vascular tissue, and provides both protection and nutrition for the gall causer. So let's just break this down a little bit. The fundamental thing to understand about a plant gall is that what you are looking at when you are looking at a plant gall is plant tissue. It has just grown under the influence of a causer. So it has grown under the influence of something else. And that organism is provided with both protection and nutrition. So if we were to go back a slide again, I'm slightly reluctant about doing this in case we get frozen again. But anyway, here we are. So let's just take the third image, which we consider is not a gall. So right at the end, the larvae of the leaf miner in there, it's a dipteran, a fly larvae inside that holly leaf, is indeed provided with protection and nutrition from that leaf. But the holly leaf has not actually fundamentally changed or grown in a different way under the influence of the causer, whereas the first 2 images, very obviously in the nopper gall, where the acorn, so the fruiting tissue of the oak has effectively grown in a way to create a totally different structure. And inside that, of course, you would find hollow chambers where the wasp larvae are living. So it needs to sort of meet a number of different criteria to actually be defined as a gall. Okay, let's jump on. So here's kind of a reason why, sorry, I can just hear someone else talking there, but so in terms of I suppose identifying plant galls, one of the reasons that I love plant galls is because many of the gall causes are incredibly difficult to identify. I've naughtily put impossible there, which of course they're not impossible to identify, but they can be very difficult. So in this image, I found this little gall midge actually floating in some water that I left out overnight once, and popped it under the microscope and realized it was a gall midge. You can pretty much tell that by the wonderful beaded antennae, also by the structure and the wing venation, very simple structure on the wings there. But I personally can't tell you any further than the family in terms of Cecidoniidae, which is the family of the gall midges. Anything more about that. But luckily, these gall midges, a lot of them will induce galls, and we can identify to species level the causa. And that's one of the great things about plant galls, is they are often vastly easier to identify than their causes. Okay, so I'm just going to wait for the screen to flick over. It seems to have this slight delay. And then we're going to talk a little bit more about the simple steps of identifying a gall. So when you're out there. So I realized from the poll that some people have some knowledge of perhaps going out and identifying galls, but I think sometimes it's actually worth going over the basics, because occasionally I do note that occasionally some people will try to skip a step, and I really would strongly advise against that when you are trying to identify galls. So step one sounds very simple, doesn't it? Identify the host plant. I cannot emphasize enough, if you are a beginner, how important this is in understanding plant galls. If you jump straight into trying to understand what the gall is, and occasionally on, for example, social media, I see, close-up images of a gall with no background information about what the plant host is, and you're basically heading for difficulty in terms of trying to sort of get towards your understanding. So it sounds obvious to say identify the host plant first, but it is essential, really. I know that there are some plant galls which are so obvious that you can see them from a distance and you would know a robin's pincushion, one of the wasp-induced galls on rose. From a distance, you could probably identify it before some people might be able to identify the plant. But fundamentally, and for the majority of plant galls, you must start with the correct identification of the host plant. And then to delve into that in a little more detail, it's really important to try to understand what part of the plant you're actually looking at and where the actual plant galls are. And you can actually get into a little bit of detail here. So you've got, are the plant galls on the leaf blade? Are they on the petiole, on the bud, on the fruit, on the flower? And also sort of trying to understand, are the galls randomly placed? Are they on leaf veins? So for example, if we look at the image on the left, so this is obviously an oak leaf, and you have here a number of immature galls. Now I've deliberately chosen this photograph because immature spangle galls are quite difficult to identify. Now when they're relatively mature, they're pretty easy to identify, and we'll see that in further slides down the line. But at a very immature level, they can be a little bit difficult. So it's important, first of all, to understand, okay, what's the host plant, where are they, and then really through some identification. And sometimes you will need to allow the plant gall to mature, and you may, of course, I'm getting ahead of myself, need to actually understand what's inside the plant gall. So yeah, we've got wasp-induced spangle galls. You've actually got two types of spangle galls, both immature on that oak leaf, caused by the wasps, Neuroterus numismalis and Neuroterus quercus bacarum. And then on the right, the two images are both on the plant Alexanders, and that's Puccinia smyrnii. So these are fungal induced galls there. So just to recap, please do identify the host plant and be sure of that first, and try to get as much information on what part of the plant you are actually looking at to understand what the plant gall might be. Step 2, again, this one can be sort of broken down into a number of different sort of substeps. Substeps is magnify, photograph, and or collect. This is obviously important. So here in the first image, you've got mature silk button galls, again caused by the wasp Neuroterus numismalis. So let's just talk a little bit about this issue of studying the galls and whether you need to collect them or not. Again, another wonderful thing about plant galls is a large number of them can be identified quite reliably in the field. If you don't know what they are, you can take a photograph of them. So those ones are the silk button gall, that's under some magnification, but taken still with my phone, just with a little lens clip on it. And, you know, I'm quite confident in being able to identify those galls without having to cut them open, take out the larvae, rear it, and identify the causa on that basis. But sometimes you do need to collect. Now, the galls on the right are, these are on alder leaves, and these are mite-induced galls. You can tell they're mite-induced galls pretty easily by the fact that there are what we call erinea patches on the underside of the leaf. So these are sort of collections of minute, differently shaped hairs, and different mite causes will cause the plant to create different shaped hairs. Now, I'm pretty confident that these galls on alder, with the discolored raised bumps on the upper side of the leaf and those particular types of erinea patches underneath, the combination of the host and the nature of the gall that we are looking at, I am pretty sure that these are caused by Acolytus, by the mites Acolytus brevitarsus. To be slightly more sure, I would pop those erinea patches under a microscope, and what you would hope to see is the shape of the hairs. I don't really know quite how to describe them, but sort of quite extraordinary looking things. These aren't sort of straight hairs, that they branch off at the end into numerous sub-branches of the erineal hairs. And that would pretty much guarantee that you know what you are looking at are caused by the mites Acolytus brevitarsus. Now, mite galls, which happen to be my favorite, by the way, are somewhat problematic in that your ability to actually study the causes themselves requires a significant amount of expert knowledge, really, of acarology, and quite significant microscopic power to be able to actually study the mites themselves. I don't have that capability. So my only real ability to understand, to learn about, and to record mite-induced galls is by looking at the galls themselves, and sometimes the galls under magnification. So I suppose the thing I wanted to just sort of emphasize to people out there, you know, there may be some people who are very used to collecting specimens and very used to rearing things and studying them. But if you are new to plant galls and you're sort of keen to get involved, and perhaps you may not even particularly like collecting specimens, you don't need to in a number of cases. I think the important thing is that you are aware that there will be cases where you need to accept you can't take it to species level unless you collect it. And I think as long as you respect that principle, it is perfectly acceptable to build quite a comprehensive list of galls that you have found simply from looking at the galls themselves without even opening them. You will, of course, get further and you will broaden your knowledge if you are willing and ready to actually explore, to magnify, to collect, to rear and study them in that way, of course. But I just wanted to sort of say that. So if occasionally, I do see this sometimes on social media groups and things where people say, you know, you need to be, you know, you need to rear that out. It's well, sometimes you do, not always. And I'm a little cautious if we're sort of putting people off thinking, oh my goodness, this is complicated study and this is putting me off. You can have plenty of enjoyment and understanding and the ability to identify a large number of galls without sort of complex study of them. So step 3, again, really sort of important, is we all need tools to help us identify things. And without keys and without illustrative guides, none of us would get very far at all. So I've already mentioned the book in the centre, which for British plant galls is kind of, if you are in any level interested in plant galls where you intend to spend any real time trying to identify them, I would say it is essential reading. And I understand that there is a new edition of this book being produced, which I'm very much looking forward to getting hold of whenever that comes out. But if you're right at the beginning level, perhaps just a photographic guide, for example, this one by Michael Chinnery is excellent just for some of the common galls that you are likely to find in the UK, in Britain. But if you are actually going to take your identification at all seriously, you really do need some keys. And this one by Redfern and Shirley for British galls is it's outstanding. It's really very good. There are other resources, of course. There's a wonderful resource online of plant parasites of Europe, Bladminides, and that is very comprehensive in its collection of information and very easy to search as well. And it's often, I'm sure, as many people find, if you have other areas of interest and study, of sort of cross-referencing things. If you're going down the keys and maybe you're getting into a little bit of difficulty, or you're thinking, well, wait a second, where am I here? It's often good to cross-refer and to use other resources. And I do a little bit of galling, well, perhaps a bit more than a little bit, in the south of France as well, where I'm lucky enough to spend quite a bit of time, although not recently for obvious reasons. And I use another resource, quite a hefty three volume book by Roscombe, and that's an excellent resource for European galls. So please do use the keys. That is kind of essential, really. And the wonderful thing about plant gall keys is they are some of the easiest to use. You really can use a lot of these keys out in the field. You don't necessarily, or you can take photos of the plant galls, bring back, have a look at the photos later and key them out at home. You don't necessarily need to collect the specimens. Be warned, as I said before, though, if you've gone out in the field and you find a plant gall and you take a photograph of it and you bring it back and you key it out, you may not be able to get past a certain point without having the specimen, but for many you can. So those really are my 3 simple steps of how to identify a plant gall. And so I just want to pause for a second and talk again a little bit about the organization of galls. Because if you take something like the keys, the keys are, the galls in here are set out by gall host, so the host plants. And that's really, as I said, from an identification purpose, that is really how you want to organize your galls. If you're trying to identify something, you need to sort of drill it down. And so, for example, if you find a gall on oak, you go straight to the Quercus section in the book. And because there are so many galls on oak in the UK, there are far more galls found on oak than any other genus of plant. And so then it is broken down even further by the particular parts of the plant that they're found on. Is it on the leaf blade, the petiole, the fruit, the bud, etc. And you're basically narrowing it down. So organizing galls by host is the best way to identify. Organizing galls by causa is really the best way to record galls, in my opinion. And that, of course, if you are recording them on an online resource, and you know, something like iRecord, and there aren't actually currently, there isn't an enormous ability to verify those at the moment, but hopefully that will come in due course. So if you're organizing them by causa, that's really how you're able to actually record the species of gall causa that you find. And the third way to, of course, organize galls is by the gall and the gall type themselves. And really, it is understanding the type of gall, and I'll come back to what I mean by the type, the structure, the formation, and it is by studying galls in that way that you will actually most advance your knowledge of galls. Now, I'm going to mention another book that isn't on here, which is, it's in the New Naturalist library. It's the Plant Galls book by Margaret Redfern, and it's an outstanding book. If you are in any level really above a beginner in galls, and you're really interested in plant galls, I strongly recommend that book. It's a pretty hefty read, but you can dip in and out of it. You know, you can sort of maybe read a section on one particular gall type and put it down and read some light fiction and then go back to it, you know, a couple of weeks later. And it really, I almost guarantee that anyone will learn something, probably many things about galls. And Margaret Redfern in that book organizes plant galls by the particular type, the level of complexity, the type of structure of the plant gall, which really does allow you to get into this area of understanding cessidology very much, you know, more profoundly. So just a quick recap: organize galls by host to identify them, organize galls by the causa to record them, and organize galls by their type and structure to really study them. Okay, so I'm just waiting for the slide to flick over, which should hopefully come through in a couple of seconds, and we'll start looking at some more galls. So as I said, I have organized in this presentation by gall causa. So starting with the family that the study of galls is named after, or possibly it's the other way around. Cecidomyidae, these are the gall midges. Remember that photo of the magnified gall midge that I showed you earlier on? It is gall midges, flies like that, that have caused these galls. And you can see different types. So the latter two images are both types of losing my words. What are they types of? They're leaf rolls. Sorry, don't know why I forgot that. They're both types of leaf roll, and the first image is a leaf fold on oak, all caused by gall midges. And this really helps us understand, again, this point about what is a gall, because a gall that is a leaf roll is very much more than some mechanically rolled leaf that you might find in the natural world, perhaps sort of glued together with silk, you know, to protect you know, larvae that are put together, and that is not a gall at all. Really, a leaf roll that is a gall, remember, the plant has adapted to roll in this way, and you are looking at sort of not complex structures in the terms of overall galls, but significantly more complex structures than simply a mechanically rolled leaf. If you took a leaf and rolled it, what you were looking at in these images is something very different. And of course, if you got inside those leaf rolls, you would find the larvae of these gall midges. And if you lifted up those leaf folds, you would find, hopefully, the larvae of the gall midges as well. I'm not going to name every single gall as we go through them. I mean, I can do if people have sort of questions later, but I think otherwise we'll sort of get a bit bogged down in scientific binomials, and, you know, it'll just sort of bog us down a bit. But remember, the other thing I'll perhaps say at this stage is that when you are opening a plant gall and looking at the larvae inside, or anything that is inside there, you're obviously hoping to find the gall causa, but you obviously also need to be prepared to find other things as well, inquilines that have parasites that have entered the gall and are benefiting, potentially feeding on the gall causas, and potentially replaced the gall causas inside. So you cannot assume when you open a gall that the things that you find in there are necessarily the causes of the gall. And again, you would then need to rear it out to actually understand what you're dealing with. And the whole study of inquilines is not actually something I've really devoted much time to at all, but it's a fascinating area of study in its own right. And you do really get you know, there are certain galls where you can almost be more, you are almost more likely to find certain species of inquiline than almost the gall causas themselves. So quite fascinating. I will actually break what I just told you in terms of not identifying some of them. This third image here, because I saw at the beginning we have so many people joining us from the US, you may recognize this gall because this is actually native to the Americas. So this is on Falsicasia, and it is caused by the gall midge Oblodiplosis robinii. And as I say, native to America, first found in Europe only in 2003, and first found in the UK in 2007. And these are ones that I have found locally to me in Wanstead in East London. So let's jump over into the rock stars of the plant gall world, not the gall midges, but the gall wasps, the CINIPIDAE. And these gall wasps probably produce some of the most incredible structures in the plant gall world. And if you're not, if you don't find these things beautiful, then perhaps plant galls aren't for you. I am often just blown away when I look at some of these structures. They're just, you know, they're so colorful, they're so intricate, they're so complicated. In, you know, look at the third image there of the robin's pincushion caused by the gall wasp Diplolepis rosae on rows. And inside that intricate mass of wiry, spiky hairs, you will find sort of a woody, very hard gall, which if you were to cut into it, you would of course find numerous chambers with the gall wasp larvae inside. The middle image is magnified common spangle gall caused by Neuroterus quercus bacarum, and then you've got the oyster gall caused by Neuroterus anthracinus on the left. Now, these oyster galls on the left, this is an example of galls that are found on the midrib of oak leaf. And what you're often looking for, sometimes you're lucky and you get incredibly colorful examples like this, but what you're really looking for is the two flaps that are only just visible in this photo that sort of surround it, and the gall comes out of it, hence its name, oyster gall. And yeah, there are very many other very colorful, very intricate galls caused by the gall wasp. So they are often, for many people, how you get into galls. I remember as a child finding nopper galls on acorns and wondering what on earth they were. They may well be people's first introduction to plant galls because they are so visible, and you're so obviously dealing with something unusual as soon as you see one that it sort of almost prompts further study. So we're very grateful for the gall wasps because they introduce a lot of people to cecidiology. So we will then flick over to not so much the rock stars, but my favorite group of gall causers, the gall mites, ERIOPHYIDAE. And, you know, as I said earlier, these gall causers are exceptionally minute, very difficult to see. You often sort of spend time when you put the aeronail patches under the microscope, as you can see in the bottom section of that first photograph. What you're seeing there is sort of you're seeing those hairs you know, actually, so these are on home oak. So this is these are Acerria illicis, sorry, that name momentarily escaped me, on holm oak leaves. And you can find these all year round because obviously we're talking about evergreen oak here. And if you zoom in on the aeronail patches, you can see these sort of thick, wiry, brownish hairs. And if you just sort of put them under the microscope and wait for a bit, you will sometimes see mites. But again, be warned, the mites that you might be looking at may well not be the gall-causing mites. And if you see a classic looking round mite with lots of legs sort of scuttling around, you're almost certainly not looking at the causer. The actual gall-causing mites are sort of often carrot, sort of elongated mite shapes where the legs are barely visible at all, certainly under my microscopic power, and moving in a very different fashion. And what you may be seeing if you're looking at sort of more classic, sort of almost like spider mites, so to speak, is perhaps something that is predating upon them. Just a quick pause here, particularly for those maybe with a little more experience of studying these galls. These particular galls, Acerria illicis, we currently count what I consider to be two types of gall as a single species. Perhaps they are, again, without really understanding the gall causes, we don't know. So in this case, you can see in the upper image of that, in the upper section of that first image, you can see those raised bumps, and underneath those small distinct aeronail patches in the depressions on the underside of the leaf. Sometimes, however, you will just find almost like a carpet of brown and very little raised bumps on the upper side of the leaf. To me, those galls appear fundamentally different, but as I say, really perhaps without much greater study actually of the aeronail patches and probably, unfortunately, of the mites themselves, you won't really be able to, we perhaps won't be able to actually make a distinction between them. And that's one of the things, if I'm honest, that I love most about galls caused by gall mites, which is that we are often right up against the current level of scientific knowledge and understanding of these galls. Now, unfortunately, in the UK, we don't appear to have any acarologists, people who study mites, focused on gall causes at all. I think there are perhaps a few experts in the US, and I believe I've certainly read some very good papers from some experts in Eastern Europe. And, you know, perhaps elsewhere. I think that there may be some in Asia as well. But, you know, it's a real shame, you know, if I sort of sometimes when I'm dreaming, you know, of maybe sort of leaving my job in finance and you know, retraining, I kind of imagine that I might study as an acarologist and and actually sort of further our knowledge a little bit. But I think that's perhaps just a pipe dream. Anyway, gall mites cause these, the middle image is of nail galls on Lyme. And, so inside those beautiful red nails, again, very, very, very commonly found, if you look at Lyme leaves, you would find a mass of hairs, and in amongst the hairs, you would find the gall mites. Again, just a pause just to go into a little bit more detail on nail galls on Lyme. This, again, underlines the importance of understanding your host. In the UK, you really do need to be able to understand the difference between small-leaved Lyme, large-leaved Lyme, and the hybrid common Lyme, because if you see nail galls on small-leaved Lyme, you can be sure of the causa. If you see nail galls on large-leaved Lyme, you can be sure of the causa. If you find nail galls on the very commonly found hybrid, you cannot be sure of the gall causa, because both gall causas can be found, and they are very difficult to identify from the galls alone. And so you can't actually take it to species level. So that's just an example of the importance of really understanding your host. And then the third image there is an incredibly tight leaf roll found on beech, and again inside that leaf roll you would find again the hairs and all the mites protected inside. And on the image, which for me is underneath all of your faces, is these are small sort of pustule galls found on elm, and again inside each pustule you've got numerous hairs and the galls inside. So those are mites, my favorite, as I said. And then just quickly to sort of zoom through everything else, I will just flick onto the next. I say quickly, and I have to wait each time before getting through. But we will look at some of the other gall causa types. But really, for me, and because I have a slight predisposition to focusing on the leaves of plants, the gall midges, the gall wasps, and the gall-causing mites are really three of the key types. I'm just hoping that the page is going to turn. Let me try again. If it's not, I'm going to have to quickly switch to my PDF. Maria, how am I doing for time? Sorry, you're muted, Maria.
It's 21 minutes past, so you've got a few more minutes. That's fine. That'll be enough to finish off. I wonder if it's worth switching over to the other option, just because this doesn't look like it's going anywhere.
I think you're right. Oh, there we go. Gosh, look at that.
Having said that.
No, so yeah, just to quickly sort of run through lots of other types of gall causa. Sawflies, also hymenopterans, particularly on willow in the UK. The hemipterans, such as aphids, cause some wonderful galls in the UK, particularly on elm, that like those balloon type galls or fig type galls. Lepidoptera cause galls. Obviously fungal cause galls. So here you've got that bottom photograph taken in my garden on my pear tree. That's a fungal induced gall. Plants can also cause galls on plants, with the most famous example being mistletoe. And then really when you are way out of my understanding level, you're getting to sort of bacteria and phytoplasms and viruses, which, because we can't necessarily identify them as living organisms, I have to say, not only do I not understand them, to me they're just particularly interested in them. But you will find things like witch's broom on things where the gall causa is again not really very well understood at all by science, and certainly not by me. So those are the other types of gall causa, and then I just hoping the page will quickly turn over. Bless you. So these are just, this is just an example of the gall causes that I found locally to me, and gives you an idea of the breakdown. It gives you a sense, perhaps due to my own prejudice, sort of around 30% of the galls that I find locally in the Wanstead area are caused by mites. Second in are the gall midges at 26%, then the gall wasps at 20%, and then kind of all of the rest beyond that. And then I just wanted to say, we are still learning. And this is the only image that I've taken that was not taken in the London area. I went to visit some friends in on the south coast of England near Poole. We went to walk to the beach, walked through a small wooded area. I found some holm oak, found, you know, Acerria illicis, as I would expect, found some other galls which I couldn't remember the name off the top of my head. I was, you know, trying to sort of keep up. It was a family walk. I was looking at them, took a quick photo and thought, oh yes, I recognize those. Took an image, took a quick photo of both sides. I knew I'd seen them elsewhere, but didn't think anything more of it. Drove home 2.5 hours, got home and realized what I was probably looking at are galls that are not yet recorded in the UK. That just gives you an understanding that these are, in my opinion, almost certainly caused by the gall midges Drymeia Lichtensteinii, which I have found in France. Now, obviously, to be sure, I would clearly need to collect a sample of that leaf. I would need to rear out the gall midge. I would need to study it. But I believe that that gall midge that I just found on that family walk is not currently recognised as being a causa found in the UK, and there it is near Poole. So anyone who's near Poole, you get in touch at the end, and you should go and find that patch of wood. And, you know, sometime when we're allowed to travel again, I will hopefully head back down there and have another look for them. So that's it. I've probably waffled on a bit too long, but I just, you know, I am passionate about plant galls, and, you know, I'm still learning all the time. Every year I develop and my own understanding further. You know, I've found, you know, in a local area in East London, in a relatively urban area, I've found well in excess of 100 galls really quite easily, without even sort of properly sort of digging down and studying the roots of plants and other things like that. It's a wonderful area to study, as I say, sort of looking across the taxa and getting out there, looking at things that don't move and fly off when you get near them. So I do encourage it. So I'll leave it there. Thank you.
Thank you very much, James. That was a really informative and useful talk. And I think you sort of fired people up with enthusiasm. Your kind of passion for galls really comes across. And I think there were people I saw coming through the chat, people going, oh, I want to go out and look for galls now, because you were kind of making them very enthusiastic. I thought it was very helpful to give a way in, and the way you'd kind of organise the sort of 3 steps and the ways of working systematically through those. I thought that was really helpful. And I kind of found the way that actually there's 3 different ways of organising the galls. You're thinking about the host plant when you're identifying, then you're thinking about the causa when you're recording, and then you're thinking about the structural formation to understand them. I had not kind of considered it in that way before, and that was really, I found that really helpful. We've got lots of questions that I've seen coming through the chat, and there may be a couple of people with hands up that would like to ask questions as well. So can we go to the chat first?
Yeah, shall I call on some people who've asked questions? So Helen Roberts, if you're still there, had a question about the colour of the galls. Helen, are you there?
Did you want to ask in person, or would you like David to ask for you?
I'll read it out.
Thank you.
She hasn't got a microphone. Fair enough. So Helen asked, Helen asked, where do the galls get their colour from? Does it come from the plant, or does it something the causa do create the colour? And a related question, and Jeremy Wright also asked why they don't keep the leaf colour to conceal the parasites.
Yeah, good question. So almost any question really about the structure and the formation really of galls and how that comes about again is is kind of really at the very boundaries of science. I mean, you can get about as close as you can understand it. For example, reading something by Margaret Redfern, really truly understanding this is well beyond my level of knowledge, and as I say, is kind of pushing the boundaries of what we understand. So to take that point about sort of color, I think, yeah, the important thing is that somewhere in the evolutionary process of, let's take, just for simplicity's sake, the case of insects which oviposit, which lay their eggs inside a plant, and there is something often from the immediate process of the ovipositing, as soon as effectively the plant is the plant vascular tissue is sort of broken in that way. The process, believe it or not, of the gall, that chemical process which induces the plant to grow in a different way begins. And through evolution, you get these sort of more complex structures to provide better protection and better nutrition for the gall causes inside, for the gall causing larvae in particular. So really, it's through that process that you have got to a point of, where the coloration comes out. I can't really tell you why that is, or how that is formed. And yeah, very good question in terms of sort of, the protection. I suppose, to go back to my other area of interest, think a little bit perhaps of, male birds coloration. The coloration of male birds is not particularly great for them to be protected from predation, but it is that balance in that evolutionary process of being able to attract, to mate, to reproduce, as well as thinking about other things like protection. So who knows, maybe others on the call sort of know more about that, but I think that's about as far as I can go with that, I'm afraid.
Thank you. I'm going to go to somebody with their blue hand up. Catriona, would you like to unmute yourself and ask your question? Catriona, are you able to do that? I've tried to do a request to ask you to unmute. Okay, I'm going to go to Angela Dougal.
Sorry, was it Cat?
Go back to Catriona. Okay, sorry.
Sorry, I keep trying to change it to Cat online, but they
right. Okay, sorry. Okay, go ahead.
I almost forgotten what I asked. Oh, it was just about seasonal. Was it the one? Unless there's another Katrina. I was asking whether it was seasonal, whether you can find these all round,
great
all
question.
year round, or.
Thank you, Kat. Yes, really good question. Yes, I mean, I suppose it follows really the development of the leaves. So inevitably, you are going to find more leaves, more galls at times when you have, for example, during the spring and summertime, undoubtedly. So as I mentioned earlier, with regards to things like holm oak, evergreen oaks, you will find galls, throughout the year. But yes, it is seasonal, and it's partly to sort of reflect the seasonal changes in the plants, but also the seasonal changes in the often very complicated sexual sort of reproduction cycles of the gall causer. I mean, just to give a quick example of that, with a lot of gall wasps, they have highly complicated sexual reproductory cycles, where you have an asexual and a sexual phase, so you have the same species causing different galls at different stages and on different hosts and at different times. So, that point around how do we organize galls is made even more complicated, particularly if you're studying by gall causer, when you're dealing with the fact that a single species in different forms can cause different galls. But yes, I mean, one thing I would say about that seasonal point as well is you can also study galls when a lot of galls, when they are vacated. And when there's nothing inside them. So for example, that beautiful image that I showed you earlier of the robin's pincushion on rose, you can see those often on rose bushes when they just look like large wooden, sort of slightly soggy masses that have been left behind. Now there's nothing inside there anymore. You won't be able to study the larvae, clearly. They've all vacated, but you can still identify the plant gall beyond that. And of course, other galls that are caused on sort of, the woody parts of plants, particularly on trees, will remain intact potentially for many decades. So yeah, there are a number of different sort of, you can study galls throughout the year, but it definitely really hots up in sort of spring, summer, autumn.
Yeah.
Thank you. We're going to squeeze in a couple more questions. Is that okay, James, if we just do a couple more?
Yes, absolutely.
Okay, so I'm going to go to the one more person who's got a raised hand. So we're going back to Angela Dougal. Would you like to unmute yourself and ask your question?
I think it might be me, but I certainly haven't got my hand raised, and neither have I got a question. My name is Dougal, not Dougal, but it might be another Angela who's
all right. No, yeah, not to worry, that's fine. Thanks for popping in and saying. Can we go to you, David, then, and take a couple of questions from the chat?
So I had one question for someone who said she didn't want to be coming and ask it herself, so I'll ask on her behalf. She was wondering whether what the impact of pollution is on gall causes and galls, and are there other environmental impacts to think about?
Gosh, yes. Great question. And I'm just trying to think whether I have any real knowledge of this at all. Anything really to say? I'm not sure that I do. I think, undoubtedly that, we see impacts, don't we, in terms of, right across entomology, in terms of, responses to outside environmental changes. We're undoubtedly seeing a spread of some galls as a response to climate change. I say undoubtedly, perhaps I shouldn't quite say that strongly, but we are seeing the spread of galls. Perhaps that was sort of more naturally be found in southern parts of Europe, found further north all the time, and similarly in the UK. And of course, the other external factor that's really important is the fact that wherever plants go, you're often taking those plant galls with you. And so you're effectively introducing species all the time. And because plants are traded around the world for gardening purposes, they're taking those plant galls with them, and you're introducing those into the environment. And I'm thinking about the mite-induced galls on the fuchsia bush outside the front of my house. That all of these things have been introduced, on the street trees in my street. I've found galls. Again, these are introduced species, and that sort of, it's spreading them around the world. So yeah, external stimuli like that undoubtedly have a factor. I'm sure there are some good papers on it. I haven't read many personally myself, but definitely an area for further study.
Thank you very much. And we're just going to go to one last question about kits, because a few people have asked it. So can we go to you, David, and you finish off with that?
Just in the interest of time, I'll just ask it, which was people were interested in what you use on your camera phone to be able to take such great pictures.
Oh, thank you. Yeah, gosh, I wish I could remember the model name, but it's basically just a little clip-on. If you were to go onto a well-known shopping website named after a large rainforest, and you were to sort of look at a sort of search on clip-on magnification lenses, I think it's sort of, it's got magnification power of 2 or 3 times. You just clip it onto your phone, and you can get it really close up to the subject and take better photos. And it costs, I think I got 2 clip-on lenses, one which allowed you to do funny things like take fisheye photos that I have no use for whatsoever, and another that allows me to get really close into gulls that I use all the time, and both cost me about £25. So well worth it. I strongly recommend it for anyone who sort of likes getting up to close up to insects, for example, when they stay still for you. Great resource. Yeah. And in terms of sort of other kit, I mean, yeah, the wonderful thing about plant galls is they stay still, you know, they're on the plant. So you don't actually need to sort of go chasing them around with a sweep net or anything. You do occasionally need to collect them, as I said, but you really, to get interested in plant galls, I would say all you really need is a guide to be able to identify them. And frankly, you can probably use the internet for some of that. So just, you know, get out there and you can have a look at them without spending any money at all. It's great.
Thank you so much, James. That's really fantastic. And I'm sorry to everybody. I know there are more questions. There's people with hands raised, but we do need to wind up now. I did see that you put your like an email address. Is it possible for people to contact you, James, if they want to follow up on anything? Is it possible to pop that into the chat, or pop that up there again?
Well,
okay.